please.
Welcome to this webinar. And welcome everyone. This is my name is John Aitchison, and I'm the editor-in-chief of um, Molecular Biology of the Cell. And as you know, American MBoC is the flagship journal of the American Society for Cell Biology, and it's very much a community journal. And it is in that spirit of a community journal that we host this webinar today. We have a great lineup. First, we begin with a keynote speaker, Dr. Margaret Gardel, and then we follow up with three shorter presentations from early career scientists who are the primary authors of exceptional papers published in MBoC over the past year. And we'll talk a little bit more about those papers when I introduce both Vincent Boudreau and Ashley Albright and Allison Langley. You can go to the next slide. I'll introduce them when we get to those presentations. The other thing that we're celebrating today are early career awards, and these are exceptional scientists or exceptional papers that were identified by our editorial board. And Zhang Dong will speak, representing an early career awardee. And there are other early career awardees that are unable to speak today, and they're listed here. You can go to the next slide, which has additional early career awardees.
Well, those will speak next webinar.
Yes, some of them will speak at the next webinar, correct? The last,
yeah, October 30. All right.
And
I think you can introduce Margaret.
Yes, that's what I'd like to do first. Thank you. So Margaret Gardel is our keynote speaker. She's the Horace Horton Professor of Physics and Molecular Engineering at the University of Chicago and director of the James Franck Institute. Dr. Gardel is a physicist who explores how living cells generate and respond to mechanical forces, how the networks of proteins that make up the cytoskeleton give cells their shape, enable them to move, and coordinate complex behaviors like division and tissue formation. Her work brings together physics, biology, and engineering to understand the fundamental rules of cellular mechanics and to inspire the new design of new active materials. She earned her undergraduate degree in physics and math from Brown, her PhD in physics from Harvard, and after a postdoctoral work at MIT and the Scripps Institute, she joined the University of Chicago, where she has built a remarkable interdisciplinary research program. Dr. Gardel's many honors include the NIH Director's Pioneer Award, the Packard Fellowship, and election to the National Academy of Science. But from my perspective, most importantly, she also serves on the editorial board of Molecular Biology of the Cell, helping to shape the future of cell biology publishing. And today, she's going to speak on harnessing AI for cell mechanobiology. And I'll hand it over to Margaret now.
Cool, thank you. I'm going to request to share my screen, just because I think let's see, now I should be able to do it. Great. Okay, so everything looks good on your end?
Got it.
Great, thank you very much. Okay, thank you very much for the introduction. In the spirit of this short talk, I wanted to talk about a topic that I'm really passionate about these days, about how do we bring in new methods for quantitative biology from our imaging data. And so it's no surprise to anybody, in this audience, but light microscopy has been an extremely powerful tool in visualizing the biological dynamics across scales, from subcellular dynamics of protein assemblies to cell motions within engineered and natural organisms to cell rearrangement dynamics, right? These are all movies that have been taken in my lab using confocal imaging across these scales. But a real challenge with these movies is how to extract information from them and to be able to learn information about biological dynamics, right? And one particular question that I find really remarkable is that biological matter has this inherent property of being able to maintain homeostasis, shape homeostasis, tensional homeostasis, cells, in response to some cues. But in other perturbations, it allows to execute smooth shape changes to allow for processes like cell migration, cell shape changes, and cell dynamics. Right. So how do we actually take these natural dynamics that we visualize to understand these kinds of processes? And I think that again, it's no surprise, like the basis of these machines that drive these cell dynamics across scales is the cytoskeleton. My lab mostly focuses on the machines that control cell adhesion, cell matrix adhesion, and contractile stresses, stress generation in the actin cytoskeleton, right? So these are the force generating and loading machines that allow cells to control and modulate their shape. And if we think about all that's been learned in these systems by many different labs, including quantitative cell biologists, has really been dissecting out these mechanochemical systems within these machineries within cells. And just in a very abstract sense, we can think about all the molecules and macromolecular assemblies working together as components that both generate force, transmit forces across these scales, forces. And the emergent result of this is that you get a collection of biological or living matter that allows it to become a larger structure, a cell at the cell or scale that can allow a cell to control and maintain its shape, and at this larger scale have some effective properties that are changing over time. So it's really not a material in the way we think about sort of compositions of atoms or molecules in physical systems. This is really a mechanochemical system with feedbacks that allow for this active and adaptive process. And this is an exciting challenge going forward, is thinking about how do we actually think about these types of materials that require these dynamical systems frameworks. And the other part about this that I just want to touch upon briefly is when we think about these collections of molecules, it is also the basis by which living matter processes information, right? So this is the heart. The cytoskeleton is the heart of where cues that come into the cell are sensed, allows this collection of biological matter to execute physiology, right? So it's not only the biophysical properties of controlling shape and dynamics, but it's also how matter is processing information. And so trying to figure out how to bring this into a quantitative framework, and to really think about cellular information processing, how these mechanochemical feedbacks give rise to active and adaptive systems, is something that I'm really interested in, right? Okay, and so this is the context that my lab studies. This is in the context of how adherent cells regulate the amount of contractile stresses and adhesion signaling to the external environment, including how they control forces at cell matrix adhesion signaling. Right, and so these are the kind of data we can collect in this. We can express fluorescent protein that tags to molecular motors within the active myosin stress fibers. We can do this to focal adhesions and watch dynamics and measure biophysical measurements of forces. But even though we can collect these data, what's really been lacking is a way to really analyze these data in ways that give us the answers, sort of predictive and quantitative ways of addressing the kinds of questions that I was just trying to articulate. And the kinds of questions that I still am bothered by, I was bothered by when I started my lab and still am bothered by, is like, how do you predict the distribution of proteins to predict mechanical properties across at the cellular scale? That is, how do you go from molecular to cellular scale emergence? How do you have systems of collections that maintain homeostasis in response to certain perturbations or allow these smooth transitions? How do you regulate through cell signaling a set point of how all these structures are arisen, right? These are really understanding the systems of these molecules at the scale of from subcellular to cellular. Okay. I'm going to go through one particular type of system that we and others have studied, which is the force sensing feedback of that requires tensional homeostasis, just as an example. So Yuli Wang initially about 20 years ago, and then Yamada, and then Mary Beckley's lab, and including our lab, had been studying a system where you think of an actomyosin stress fibers that's undergoing tensional stresses from actomyosin motors, actomyosin interactions. And if there is a stress that's too large, so the stress fiber starts to break, there is a force-sensitive feedback that occurs via the limb domain protein known as zixin that facilitates actin stress fiber repair. Right, so we have characterized this force-dependent feedback and looked at the molecules that are associated with this force-sensitive feedback. And Greg Alushin's group has now reconstituted this process, right? So each individual group can go through and really dissect all these feedbacks. And if we just summarize the kind of feedbacks that we get, right, we get this notion that there's a property of stress fiber mechanics, but the way you control the mechanical properties at the stress fiber scale is through this force sensitive response or repair function that's happening through the of zixin. And we seek ways, and so when we think about physical properties that are emerging through cytoskeletal assemblies, right, you need to understand all these feedbacks. But yet we still lack ways of sort of precisely characterizing those. Okay, so, and this is a challenging problem, right? We want to understand these systems. We have these biophysical or cell dynamics measurements. How do we build models that capture this? And this has been a challenge in model building. There's lots of length and time scales. There's really unknown mechanochemical feedbacks. We can never measure these parameters. So another way of thinking about this problem is that it becomes a problem. How do we reduce the dimensionality of our problem? You know, traditionally, when we build cell biological models, oftentimes we start to build molecular-based models where we try to model all the biochemical interactions in the system, model every component to build and to make a prediction about the emergent response. On the other hand, traditional physics would say like, oh, we just have a coarse grain approximation of a problem like pressure or stress. And we have a few number of parameters that have no molecular identities. But this is sort of gives you a continuum theory, which we can now predict features of. But what's happening when we try to model build in cell biology is we really have struggled to go between these different scales of model building. And this becomes a dimensionality reduction question, okay? And so the exciting thing is that AI methods to analyze our imaging data give us a really new powerful tool to do this dimensionality reduction. So I just want to describe in a few minutes the way we started doing this with our biological imaging data. And this is a collaboration that occurred between Patrick Oakes's group at Loyola and my group, and Vincenzo Vitelli's group at Chicago, and our groups that all work to collect the data and then develop the AI methods to analyze these data sets. And so the question we posed was, if we take an image of a fluorescent protein, in this case it's going to be zixin, and then we make a measurement like a biophysical field, like a traction force measurement, can we build a neural network, a convolutional neural network that does an accurate prediction of the image of zixin to this biophysical field? And the exciting thing is that even with data sets that a single person in my lab or another person's lab could collect within a week or two, you can robustly train a convolutional neural network to train a model, which suggests that all the information that's in this zixin image is sufficient to predict a biophysical field of traction forces, that the information content in this image is sufficient to make a prediction about the other field. Right, and so we can test this. We can say, like, when we have a training set of data, can you make an accurate prediction that's as good when you have an image that was not in that training set? And the exciting thing for those of us that work on, say, tissue culture models rather than other developmental systems is that when you even train the model and then make a very strong perturbation far outside of the training regime, the model still does well. So this is an example where we hit the cell with Rho kinase inhibitor and really knocked it out of a training regime, and it still does a robust job. And the sort of the bottom line of this is that my hypothesis is that in say tissue culture models that have all this variation, we've typically worked very hard to minimize the statistical variation between our cells because we thought of that as biological variation. But what I would guess is that what these AI methods are doing is actually showing us that statistical variation is sort of showing us a landscape of which these cells are exploring. And so all that variation we see in our data now becomes a superpower, in sort of building these predictive models rather than a bug. And another way of saying that is if you have a beautiful canalized developmental system, it's really hard to build a robust ML model, whereas it's easier in these sort of messy systems because of all that variation. Okay. And so then you can start to ask questions in these systems of like what features in the image and what kind of protein, feed distributions are important for the feature that I want to predict. So in this case, you can ask like, does the outline of a cell actually give you as much predictive power as knowing all the protein distributions? And you lose a little bit, but you don't lose much, right? And that is consistent with our understanding of what properties of the cell are controlling force distributions. But say if I make images of mitochondria, it does a really crappy job, right? Which is because mitochondria distributions, the distribution of mitochondria in the cell is really not predictive of the force field, right? So you can start to now precisely ask information about what content in the images, what features of the fields that I'm measuring are predictive of the phenomenon that I care about. And once you have this, now you can use this as a tool for simulation. So now I have a trained neural network that is trained on experimental data, and now I can explore a realm of sort of make synthetic data of my protein distributions and ask what kinds of, like in this case, what kind of force production would I imagine, right? And this allows you to run through using this new type of simulator based on imaging data, which the simulator is built on our data, experimental data, and ask, in this case, what are the features of the adhesion sites that are most predictive of the force, right? And what this allowed us to do is very quickly through the synthetic data, pick up a feature of the shape and the morphology and the distribution of where the adhesion site was located to understand that was a site of maximal force, which is exactly consistent with where like a decade of experimentalists trying to measure these correlations in our experimental data also found it, right? So this is a way to rapidly iterate our experimental hypothesis building to test interesting new directions. Likewise, you can start to learn precisely these mechanochemical feedbacks. So this is an example where we can now ask, how does the force depend on the intensity of the zixin, and ask when that mechanochemical feedback is changing depending on the location and site of the adhesion. So this is a really exciting advance, and I think it's really going to be powerful for doing interpretable understanding of cell biology, especially for those of us working on imaging data, right? So it's a way of building a model that is really closely and built on our experimental data sets that we've been collecting in the lab. And if we think about sort of like the canonical physics theory, it might be interpretable, but there's a big gap between what we measure experimentally. So these neural network models really fill nicely. Of course, we want to fill in these gaps. I don't have time to talk about that right now, but we worked on ways with Vincenzo's group to like really build other ways to make more physically interpretable neural networks, but also augment physics theory with known biochemistry, so that you build a whole suite of different types of model building using data-driven approaches. And this gives you a whole new class of data-driven biophysical modeling that I think will really help with our understanding of cell dynamics across scales, using data sets that we have gotten really easily capable of doing over the past decade. Right. And I just want to end with, like, I grew up in a time when the sort of the paradigm of quantitative cell biology was this measure, manipulate, model paradigm, where you make a measurement, you build talk to a theorist that builds a model, you measure something from your data. And this is sort of blowing apart this paradigm, because there's a whole new way of model building that bridges between interpretable models and these data-driven models, which sort of live between full interpretability but are really data-driven, and to understand the physics. And I think it'll be really exciting in the future to understand how all these different feedbacks can be built to advance our understanding of quantitative cell biology. That's it. I just want to thank my group, and I've highlighted a few people that really are the collaborators that drove the work that I talked about today. So thank you.
That's wonderful, Margaret. Thank you. Irong, do we have time for questions now?
Yeah, give Margaret 3 minutes for question. I see Wallace have a question. Wallace, you wanna speak out?
Go ahead, Wallace, if you've got audio.
I see it in there. Okay, so he says, given that there are lots of proteins in the actin cytoskeleton, but only one traction field, can you go backwards and predict all the actin cytoskeletal protein distributions just from a single traction field measurement? Then it wouldn't be necessary to image all the proteins. That's an awesome question, Wallace. We don't know yet. So I think this is one of the things that I'm currently interested in is like, say with focal adhesions, they change in biochemical composition while they're changing mechanics. We have lots of evidence that the mechanical output, that is the force that they're under, is not reflective of all the signaling, but we sort of want to separate them out. Is like, what are the fields that are most predictive of the mechanical field and which ones are important for chemical signaling? Even be able to answer that precisely would be very satisfying to me. I mean, another way of asking your question is like, with all these multiplex imaging modalities, like, could we just make a big prediction of the cell state from a phase contrast image, right? Which I think is a really extreme version of asking these kinds of questions, is like, really what information in the cell is necessary to make a prediction about anything you care about. So it's a great question. I don't know the answer to it yet, but I want to figure out in the context of focal adhesions at least.
So Margaret, I think in a similar vein, one of the advantages of models is that they're often wrong, and they help us to design experiments that will make them more precise. So in neural nets, in this analysis, can you identify missing parameters that would help design the next experiments to improve the models? And how do you do that with a neural net?
Yeah, so I think, okay, so there's, I would say that my experience with the model building with the neural nets is still at the stage where I don't even trust it enough to study a question that I have really good experience with. I'm being completely honest right now, because like, we found a lot of ways that when we started trying to actually learn parameters, that you can overfit your system because you basically have all these freely adjusting parameters. So this is a challenge, right? With any of these approaches. I mean, there are people trying to do this in the case of like imagine you're trying to learn Kepler's laws from just looking at the motion of planets, and you could completely overfit your system, right? So I don't know what the answer is going to be. A cell is way more complicated than learning Kepler's laws. So that is a field of development. Okay, like that's the stage where we're at here.
Right.
Yeah.
And so similarly, one needs to identify those parameters that are mutual information, right? That are just
That's right. So
reinforcing
that's
and
another a way of doing that, which I think is really interesting with the unphysically interpretable neural networks, like taking an information theory approaches and trying to understand, of my imaging fields that I'm collecting, which ones are actually important for the phenomena I care about. That's a completely different approach that traditionally I don't think that has been merged with sort of these interpretable physics theories, but I think it's going to be really interesting to do with these kinds of approaches. With just the trained neural networks, I mean, that's a completely black box predictor, right? So I think that one, if you have a trained neural network that you've done on your data set, you can then go and ask in lots of different conditions to see whether the training regime that you did is sufficient to be an accurate prediction in another condition. And I think that is basically at the point where people could start using it out of the box. Like another lab could use our convolutional neural network that's been trained on our images and ask, does it work for them? Right? Like, I think we could start using those sort of black box approaches now and start to build some intuition about when they're actually useful as a community to get knowledge rather than individual labs.
That's great. Are there any other questions? I think you can put your questions in the chat if you have questions for Margaret.
Yeah, put in the Q&A, and then in the end, let's say we can do group question, right? Let's
Sure,
proceed. Proceed with Ashley, Vincent.
That's great.
Yeah,
Great,
Vincent.
thank you.
Thank you very much, Margaret. Um, so each year, MBoC honors one paper that truly stands out. A study that captures the spirit of discovery, rigor, and creativity that defines our field. And the MBoC paper of the year is chosen by journals editors from among the most many outstanding articles published over the past year. It recognizes work that not only makes significant advance in understanding how cells are built and how they work, but also inspires a new field for the broader cell biology community. The selection process for for the paper of the year is highly competitive and collaborative. Um, Associate editors, monitoring editors, the editorial board nominate and vote on papers that exemplify this mechanistic insight and innovation that we're trying to promote through MBoC. And the result is a recognition that it reflects the collective judgment of the cell biology community itself. And I'm pleased to say that this year we were unable to distinguish between the top two papers. And so we have the honor of being able to recognize two MBoC papers this year. The first we're going to talk about is a presentation from Vincent Boudreau and Ashley Albright from the University of San Francisco. The title of their paper was The Cell Biology and Genome of Stentor pyriformis, a giant cell that embeds symbiotic algae in a microtubule network. The study reveals how a single giant cell organizes and maintains symbiosis with algae and offers new insights in cell architecture and evolution. And Vincent and Ashley are going to do a little tag team presentation, and I'll pass it off to them right now.
Thanks a lot, John. And I'll start by thanking ASCB and MBoC for the recognition of this work. It's an honor to receive it. This work has been a labor of love, as you can see, having been worked on through a variety of co-authors here, which I'll thank as well, particularly Wallace Marshall for his supervision at the University of California at San Francisco, and Chris Niogi, I was a joint postdoc with at the University of California, Berkeley. And I'll start by setting the stage as to how we started to work on Stentor pyriformis as this giant cell and develop it as a model system for studying algal symbiosis. This work was initiated in the context of an intensive research course at the Marine Biological Laboratory called the Physiology course. This is a course in which graduate students and postdocs visit the marine biological lab. And what you can see here in this image are students being guided by Mark Slobodnik on the right, who I know is in the attendees here with us today, in collecting samples from a freshwater pond that is located behind a retirement center in Falmouth, Massachusetts, taking these organisms back to the lab and using state-of-the-art equipment to be able to observe them. It's remarkable how even in a few milliliters of this water, the amount of unknown biology that exists there. If we look more closely just at the surface of the water, and this is a picture taken simply with my phone looking down at the pond water, you can start to see what I'm talking about. There's an accumulation of these green cells here, which I'll tell you more about in the coming slides and detailed in this paper, that accumulate near the surface of the water column, particularly during the day when the sun is out. So we took these back into the lab, excuse me, and started observing them under fluorescence microscopy, something that hadn't been done yet with these cells. Initially, we were interested in Stentor as a giant single cell, and you can see the scale bar here. These cells can measure up to half a millimeter in diameter. And we were interested in using this system to study how the cytoplasm mixes in this very large volumetric cytoplasm. We thought that it might be useful to use these endosymbiotic algae as markers, basically as fluorescent beads within the cytoplasm, to be able to study cytoplasmic flow within these very large cells. And to our surprise, when we started to put this organism under a microscope and look more closely at the endosymbionts, what we noticed is that, in fact, there is some symbionts that are subject to cytoplasmic flow, as you can see in the middle there. But there are also algal endosymbionts that are constrained near the cell's cortex. So we started to look more closely at the cell cytoskeleton, and of course, looking at the microtubule cytoskeleton first. On the left-hand side here, what you can see is a cell fixed for and stained for beta tubulin, and you could start to see a pattern of microtubules that tether the endosymbiotic algae beneath the cell cortex. We wanted to look more closely at this microtubule cytoskeleton, and we used expansion microscopy to physically blow up the sample to gain resolution into the system. And as we go deeper into the cell, what you can see appear here is a microtubule meshwork that is responsible for anchoring these algal endosymbionts immediately beneath the surface. If we do a maximum intensity projection of just a few of those slices, you can see this here. And you can begin to see microtubule filaments that are part of this microtubule meshwork, the specialized microtubule meshwork beneath the surface of the stentor, which serve to tether algal endosymbionts beneath the surface of the cell. We then started to characterize the biology of this system with the goal of characterizing why or how these algal endosymbionts are tethered through this microtubule meshwork and what their function might be at that particular location. There are two behaviors that I'm going to describe here with you today, and I encourage you to take a look at the paper and the published data there, as there's more information related to this in the actual publication. The system provides a really interesting lens into interspecies communication and how two completely different eukaryotes work together to accomplish really complex cell biological functions. The first at the top here, and I'll loop this movie here, is phototaxis. So the algal endosymbiont is, of course, a photosynthetic organism within a non-photosynthetic host. If you shine light from the left-hand side of the dish at the top there, you can observe cells swimming towards the light source in the direction of light. We characterized this behavior and showed that it was chlorella dependent. So the algal endosymbionts are responsible for guiding the host cell towards the light source. We described the circadian rhythm dependence of this behavior. These cells only do phototaxis during the day and don't do this at night. And we also characterize the wavelength of light to which this phototactic behavior is responsive, and it is responsive to blue and green light. Conversely, in the bottom here, we also characterize how the host mediates the metabolic activity of its endosymbionts. I won't get into the details here due to the lack of time, but what we characterize here in Chris Niogi's lab is the photoprotective ability of this organism, which is the mechanism by which photosynthetic organisms dissipate excess sun radiation as heat. And what you can see on the left-hand side here is that the symbionts that are within the organism, as opposed to outside the organism, grown aposymbiotically or ex situ, in the host, there's a strong photoprotective mechanism in high light. What we're able to show in the organism that's ex situ here is that this behavior can be recapitulated when the organism is nitrogen limited. And nitrogen limitation is known as a mechanism for hosts to manipulate their endosymbionts' behavior. So what we're able to show here is that endosymbiotic chlorella, when they're within their host, exhibit a robust photoprotective mechanism, that this seems to be nitrogen dependent and regulated by the host's metabolism. And with that, I'm going to hand it over to Ashley, who'll tell you more about the genomics work that we've done on both the Stentor host and its endosymbiont.
Thanks, Vincent. So as Vincent just told you, the presence of the endosymbiont clearly affects the biology of the host and vice versa. But we actually, in Wallace's lab, work with another species of Stentor, Stentor ceruleus, which does not have any algal endosymbionts. And we already know that the endosymbiosis affects the Stentor biology, but as we continue to investigate this further, we wanted to know if there were any differences within the genomes between the two Stentor that could lead us to additional clues for pathways to look at and such. So I'm just going to give you a very broad overview of the differences between the two genomes. And so What we see on the left-hand side is the difference between the contag length. So it's not quite a chromosome level assembly yet, but what stood out to us here is that the contag length in pyriformis appears to be oddly consistent, which isn't really something that we or other people I've talked to have ever seen before. But what I also find interesting on the right is that if we look at the GC content for each of these contigs, the piriformis in light blue appears shifted to the right to a higher percent, which is what we see in the chlorella endosymbionts. And next slide. And then another interesting thing that I want to point out, not just because of the difference between the biology in general, but also for people who are interested in RNA and intron processing, Stentor ceruleus, so the non-endosymbiotic species, actually only has intron lengths of 15 or 16 base pairs, but for whatever reason, the Stentor piriformis introns, they do seem to peak around 15 or 16, but they have a broader range. And we think that some of these differences might actually be things that are important for this endosymbiotic relationship. And so this is something that we're going to explore further in the future. But also, if we look on the right for these benchmark orthologs, which are normally used to assess genome completeness and quality, if you look at the darker blue bars here, the Stentor piriformis has many fewer duplicated genes compared to our other strain, Stentor ceruleus. And if we look broader in the ciliate lineage, it seems that organisms with endosymbionts seem to have avoided whole genome duplication. And on the bottom right, I just want to point out that we also have a genome for the endosymbiont as well. And so all of these things together, we're going to explore computationally and then use that to inform future perturbation experiments. Next slide. And with that, we'd like to I'd like to thank Vincent first for being so fun to work with, and also Wallace for being a wonderful advisor, and also thank the rest of the co-authors on the paper, as well as our funding sources and ASCB.
Thank you, Ashley and Vincent. We can have more questions later, but perhaps are there any questions we can address now?
I
I
did
have
not see the
have
Q&A. Yeah.
I have one if you don't mind. I mean, it seems to me this is a fantastic example of, evolution in process. And I'm wondering if you could put this on an evolutionary timescale, this endosymbiotic event, and whether we know, or whether Stentor pyriformis can survive without the endosymbiont.
Yes, so I'll address that from multiple different angles. We've been able to isolate the endosymbiont from the host. The endosymbiont can survive on its own, and we've grown it and cultured it, and it's available now publicly as a strain. The Stentor host can survive for some amount of time without its endosymbiont, and by that I mean hours. It's unclear whether or not it's just missing a metabolic factor, like a food source, for example, to be able to survive without its photosynthetic partner. So that's the extent of the experimental work that's been done there. I think the more compelling argument is actually on the genomic side. And interestingly, as Ashley pointed out, we were not expecting this, but the host genome is actually quite small in comparison to other aposymbiotic ciliates. So conversely to what you might expect, for example, between a eukaryote and a bacterial endosymbiont, for example, a mitochondria or a chloroplast, which end up having genome reductions that are of significant degrees, this is actually the flip side, in which the host genome seems to be, quote, simpler in nature, smaller, with fewer gene duplications, as opposed to its endosymbiont. So it's an interesting question that we'll explore further.
I mean, Ashley mentioned, genome duplication, that event that is that has not occurred and is typical in these other large cells. But I'm wondering if are there molecular paralogs or anything like that that we can see between the two species?
We haven't done a deep dive in between the two yet, but I do want to point out regarding your evolutionary question, there are other species of mixotrophic ciliates, one being Paramecium bursaria, which also actually has the same species, the Chlorella variabilis endosymbionts. And to me, it seems like these endosymbiotic species are at totally different points in the ciliate tree. But I think one thing that could be really exciting is to go back to Woods Hole. And there's actually in one of the ponds, there's both this Paramecium bursaria and Stentor pyriformis that both have this presumably the same endosymbionts. And I'd like to look at the differences between these isolated from the exact same place in the wild.
Can I come with you? That sounds
Yeah,
fun.
sure. It
Okay.
is fun.
Yeah, we have one question from Ross. Just a very brief answer, and then you can type detail in the chat. Okay.
Okay. In the chat?
Yeah. Vincent, you see the chat, Ross question?
Yes, I do see it now. So the question is, what fraction of Stentor genes have introns? Is it a very low fraction like in Saccharomyces cerevisiae or higher like in metazoans? The intron composition in Stentor piriformis is quite different from Stentor ceruleus. And I want to say that most genes in the piriformis genome have introns and they're of more variable length as opposed to in ceruleus, which is clustered around very short intron lengths.
Thanks, Vincent. Thank you, Ashley and Vincent. And we'll move on to the next presentation. The second paper of the year honoree is presented by Allison Langley from the University of Vermont. This elegant work uncovers how the ER uses diverse vesicle systems to move and reorganize within the cell. It advances our understanding of dynamic organelle behavior. The title of her talk today is "The Movement of the Endoplasmic Reticulum is Driven by Multiple Classes of Vesicles Marked by Rab-GTPases." And I believe, there it is. Yurong has put both papers now in the chat where you can take a look at them directly. And Allison, I'll hand it off to you.
Okay, hi everyone. My name is Allie. Can you just give me a thumbs up that you can hear me?
Yep.
All right, great. So I am a grad student in the Salgy Honest Lab at University of Vermont, and I'm going to be telling you about our work on the endoplasmic reticulum today. I'd just like to start by thanking MBoC and ASCB for having me. It's really a great honor to be recognized in the community, and I'm really excited to be here. So as many of you know, the endoplasmic reticulum, or ER, is a large organelle. It extends throughout the entire cell with a variety of functions like protein folding, protein synthesis, and calcium homeostasis. And interestingly, the ER tubules in the cell periphery are highly motile, as you can see here. And all of these movements are occurring on microtubules. And there are two kind of canonical ways that we think about ER movement. The first being sliding, where ER tubules associate with a molecular motor, and then that motor can pull the ER tubule along microtubules. And then TAC, or tip attachment complexes, where the ER associates with the plus end of a dynamic microtubule. And then as that microtubule grows and shrinks, the ER tubule grows and shrinks with it. A third, newer mechanism that we're starting to appreciate is called ER hitchhiking. And this is where an ER tubule will associate with a motor-associated vesicle like this one here via a membrane contact site. And then as a motor transports that vesicle, the ER tubule gets pulled along behind the vesicle. And work from a few labs, some of whom I've listed here, have shown that these vesicles are endolysosomal, meaning that they're marked by Rab5 and Rab7. So this led us to wonder if other classes of vesicles might also be participating in ER hitchhiking. So we screened through Golgi-derived vesicles, and we landed on Rab6, which is a major class of Golgi-derived vesicle. And as you can see, these Rab6 vesicles shown in green are able to pull these ER tubules shown in magenta around the cell. And we quantified how frequently this was happening, and we found that almost 20% of all ER movements were occurring via this Rab6A or B pulling mechanism. And we also found that this Rab6 mechanism was entirely independent of the previously described endolysosomal ER hitchhiking that I mentioned earlier. So as cell biologists, we love asking questions of necessity and sufficiency. So we wondered if Rab6 movement is necessary for ER movement. And we started by making a Rab6AB double knockout U2OS line by CRISPR-Cas9. And then we quantified ER movement in these cells, and we found a significant reduction in ER movement overall in the cells lacking Rab6A and B. And this indicated to us that Rab6 movement is indeed necessary for ER movement. So we dug a little bit deeper into this question of necessity by inhibiting, instead of just completely deleting Rab6 from the cell, we inhibited only the ability of Rab6 vesicles to move. We did this using the Rab-log heterodimerization system. So we fused FKBP to a Rab6 vesicle and then FRB to a Riger mutated kinesin. So this kinesin is not able to walk along microtubules. It just binds the microtubule and sits there like an anchor. And then upon adding the Rab-log heterodimerizer drug, the FKBP and FRB will heterodimerize, and this will link those Rab6 vesicles to the microtubule anchored kinesin and should stop the movement of the vesicles. So I'll show you an example of what this looks like in a cell. These are Rab-marked vesicles shown in green, and you'll see a blue circle when the drug is added. So as you can see, adding the drug and linking the vesicles to that microtubule anchor really does stop the vesicle movement in their tracks. And when we quantified ER movement in these cells, here I'm showing you a graph where connecting lines indicate the same cell pre and post addition of the drug. And we found a significant decrease in ER movement when we added the drug and linked the vesicles to that microtubule anchor. And this is in contrast to cells where we treated them with an ethanol vehicle only and saw no change in the ER movement, indicating that Rab6 movement is indeed necessary for ER movement. So next we moved on to the question of sufficiency. Is Rab6 movement sufficient to drive ER movement? And we found, so we again used the Rab-log heterodimerization system, but instead of using a microtubule anchored kinesin, we used an active kinesin. And the hypothesis was that adding the drug and linking these Rab6 vesicles to the active kinesin would result in the vesicles and potentially ER being transported out to the microtubule plus ends in the cell periphery. I'll show you what this looks like in a cell again. The Rab6 vesicles are green, ER is magenta, and then you'll see a circle when the drug is added. So as you can see here, adding the drug and linking the vesicles to those active kinesins causes both the vesicles and the ER tubules to move en masse out to the cell periphery. And again, we saw no change in ER distribution when we treated the cells with ethanol alone. So this indicated that Rab6 movement is indeed sufficient to drive ER movement. We also stained the cells for ER tubule marker RTM4 and sheet marker CLIM63, and we found that the majority of these ER rearrangements in response to the drug were RTM4 positive, indicating that they are indeed ER tubules that are rearranging, not ER sheets, and that these movements are occurring on microtubules, because when we treated the cells with nicotazole to inhibit the microtubules, we saw a significant reduction in ER movement when in response to adding this drug. So overall, these data indicated to us that Rab6 movement is indeed necessary and sufficient to drive ER movement. So in this paper, we were able to identify that ER tubules are able to hitchhike on these Rab6 vesicles, and that Rab6 vesicles are both necessary and sufficient to drive ER movements. And that this is a completely independent mechanism of previously described ER hitchhiking on endolysosomal vesicles. And so kind of where to next is that one of our lingering questions here is what molecules are tethering these ER tubules to these Rab6 vesicles. And we have a grad student working on this now, so hopefully you all can read about that in the future. And I think understanding the tethers will give us insight into the overarching biological function of ER hitchhiking, which is still unknown. But I think it's definitely possible that many different classes of vesicles are at play here. And this would create an interconnectedness between a variety of signaling pathways and overall ER dynamics. And I think that provides a really exciting avenue for future research that I'm excited to get into in the future. So with that, I would just like to thank MBoC and ASCB again for having me. And I'd like to thank my lab, as well as my department, program, our funding. And I'd like to give my greatest appreciation to Sarah Abling-Wong. She is a tech in our lab who was co-first author on this paper, and she was an absolutely integral part of this work. And with that, I am excited to hear your questions.
Thank you, Allison. We can, as Yurong has asked that we attendees put their questions in the Q&A, and after the next talk, we could have a few more minutes for grouped questions. However, I have a quick question that I'm going to take my prerogative and ask. So when you delete Rab6, do you see also a collapse of ER tubules to the interior of the cell away from the periphery?
That is a good question, and that's not something that we quantified, but we definitely could. So I will say by eye, we actually don't see like a collapse of the ER. And I think part of that might be because there are other, there's these other movement mechanisms at play that might be kind of compensating. But it is definitely something that could be quantified that we could look into. But by eye, not really.
So you think of this as one of perhaps several mechanisms, and there are more redundant mechanisms to discover? Is that how you think of it?
I think maybe. I mean, so we know for sure that sliding and tack are at play. And then we now know that these endolysosomal vesicles are pulling ER tubules. Rab6 is pulling ER tubules. And we also have some data, which I didn't have time to talk about today, that Rab1 might be pulling some ER tubules. This is not something that we focused on a whole lot in this paper, but it's definitely something that could use a deep dive. I also saw another question about Rab11. We did quantify if Rab11 can pull ER tubules, and we saw very little co-transport between Rab11 and ER tubules. So yeah, I think, and those are just the Rabs that we've studied. So I think there are potentially a lot of different organelles that could be modulating ER dynamics that have yet to be discovered.
And one other question in the chat was, what happens when you overexpress GTP locked Rab6? Have you done that?
Yes, we did do that. And we actually see no difference between overexpressing a GTP locked Rab6 versus a wild type Rab6. And there's no difference between those and just having endogenous Rab6. So there's no overexpression phenotype. We do see a reduction in ER movement when we express a GDP locked Rab6, dominant negative, but the constitutively active GTP locked has no effect.
Great. Thank you. Thank you, Ashley. We'll move on to our next speaker. In the process of evaluating and looking at some of these exceptional papers, we also identified, or the board identified, several early career awardees and their exceptional papers. And so we did plan for 3 speakers in this group, but unfortunately, 2 of our MBoC early career awardees had to drop out at the last minute. But we recognize them all, and we'll get back to that. We'll show that slide again at the end as we wrap things up. But our speaker is Zhang Dong from Harvard Medical School, and she's going to speak on the paper STMND1 is a phylogenetically ancient staphylin which localizes to motile cilia and exhibits nuclear translocation that is inhibited when soluble tubulin concentration increases. This work uncovered a conserved connection between tubulin dynamics, ciliary function, and nuclear signaling, and reveals new links between cytoskeletal regulation and cellular communication. And so we'll hand it over to you, Jean. You're muted still, and I think you can share.
Yep.
Yep, we see it now. Thank you.
Yeah.
Wonderful. First of all, I'd like to thank the editorial board of MBoC for this award. The community built around ASCB is both welcoming and nurturing, and I'm deeply grateful to be part of it. And it's my honor to present today. I know the title of my paper is probably quite the longest, and you might wonder why I became interested in this specific protein. In short, I characterized a new tubulin binding protein called stathmin D1. By studying it, I showed that soluble tubulin can actively regulate the location of stathmin D1, not just serve as a building block of microtubule. What are stathmins that we're talking about? Stathmins are the proteins that share this tubulin binding domain. It's called SLD. They are linked to many human diseases like leukemia, neuroblastoma, and neurodegenerative disorders. So at the time, I had been studying all the other stathmin family members, but it was really hard for me to pinpoint the exact functions of them in living cells from a microtubule tubulin centric point of view. The model just cannot cohesively explain what I've observed. So I decided to look at the evolutionary history of stathmins and see what their ancestors did. By searching in the database, and I found this group of proteins. It has a gene name called STMD1, and it has two features. One is the motif that can be myristylated and palmitoylated, leads the protein probably to some membrane. And there is another feature that there is a nuclear location signal lies right within the SLD domain, which is supposed to bind to tubulin. So these two features, a unique feature also shared with the very first SLD containing protein that I identified from choanal flagellate. So I got interested in this protein in the relationship with the evolutionary history of the stathmin. So I collected all the representative stathmin SLD containing protein sequence and did phylogenetic modeling. And here's what we see that stathmin D1 actually indeed is very close to the choanal flagellate stathmin, comparing to all the other conventional stathmins that people are talking about. So the interesting question is, are those computational predicted features real in cells? So I expressed stathmin D1 along with the mutants of stathmin D1 in generic U2S cells. And you can see here that the N-terminus, those myristylated and palmitylation motif, indeed can lead stathmin D1 to the membranes, whereas the SLD region containing the nuclear location signal will guide stathmin D1 to inside the nucleus. And Brian and I, the graduate student from the lab, purified the protein, and we did analysis in biochemical analysis in vitro, and we found that the binding of stathmin D1 still, although with that nuclear location signal within the tubulin binding domain, it can still bind to tubulin with a similar binding affinity comparing to other stathmins. And we're curious about what activities stathmin D1 is involved in relative to tubulin, since it's a tubulin binding protein. So we combined proximity proteomics with a drug treatment strategy using microtubule stabilizing drug and microtubule destabilizing drug to manipulate the soluble concentration, the concentration of soluble tubulin within the cells. And what we found is that when the soluble tubulin concentration increases within the cells, plasmin D1 binds to tubulin, whereas when the soluble tubulin concentration decrease and the plasmin D1 largely enter the nuclei. And this discovery is also has also been confirmed by live imaging. We expressed the fluorescent tagged protein and look at the intensity within the nuclei and also in the cytoplasm. And then you can see here that the fluorescent intensity increases when the cells were treated with microtubule with paclitaxel, the microtubule polymerizing drugs, and the intensity decreases when the cells were treated with microtubule depolymerizing drug. Information from the database also indicated that plasmin D1 gene is expressed in cells with cilia. So we went on to stain human tissue with both ciliated cells, both primary cilia and motile cilia, and we found that plasmin D1 is enriched in those motile cilia in human tissue, but not primary cilia. And the location of the plasmin D1 indeed resembles the traits of the ancient plasmin that we found in choanoflagellate. So the interesting question would be what kind of ancestral function that has been preserved into even human, from choanoflagellate to human body, and what is tubulin's role in all that. More work needs to be done, but our discovery that excites me the most is that conceptually, we think tubulin is not just a building block of microtubule. It can actually regulate from our case, regulate the location of STMD1, and actually leads me to other discoveries of plasmins that their location and function has also been regulated by tubulin. With that, I'd like to thank our collaborator, Professor Marian Koloksi and Professor Sandro Santaga. And I have deep gratitude to show to my mentor, Dr. Mitchison. Yeah, that is our lab, and I'd like to thank our funding source and the MBoC and ASCB. Thank you.
Thank you, Jean. I'm going to do a quick look in the chat. There are no questions there yet. Maybe I could ask if you could expand a little bit on what proteins plasmin D1 binds in the nucleus, and whether you can have any hypotheses about what it's doing there.
Yes. So our hypothesis is that plasmin D1 is probably involved in RNA processing. From the proteomics results, we see a lot of RNA binding protein. And also, if we once the stasmin D1 enters nuclei, it mostly enriched at nucleoli, which is the hub for RNA processing, RNA splicing. And also when we overexpressed stasmin D1, the morphology of nucleoli changed. It's almost became hollow and disformed. So we think either stasmin D1 helps maintain the structure, but it can also, like the stasmin D1 would affect certain RNA for gene expression in that size, because what we think is that stasmin D1 enriched at motile cilia, and the cilia, the primary function of cilia is to sensing the environmental signal, and the environmental signal is augmented within the cilia. So a little bit change of external signal can be interpreted to internal intracellular function. That is our hypothesis. And at the moment, we lacked the model to really study the biological function of stasmin D1. In the paper, we reported that expression stasmin D1 changes the length of the cilia, but we used a primary cilia model. And that is not ideal. So in the future, we'd like to use some animal models like zebrafish to really study the motile cilia and study what STMD1 is doing. Yeah.
Cool. Very cool. And do you think the lipidation on the N-terminus is regulated? Does it come on and off? Do you know?
Yeah, I think so. I think it's an active. I think it's an active function. Like the membrane binding domain, which is by myrcillation and the palmitylation, comparing to the common ones. It is this kind of modification allows that the protein to become on and off from the membrane, and also this motif can be cleaved. So it can also, like, the protein once the motif being cleaved, the protein will be off membrane and they won't go back again, and that is another possibility.
Okay, there are 4 questions in the chat that I didn't see until now. Some overlap with questions I asked, but let's ask them anyway. Does stasmin D1 have any role inside the nucleus? We discussed that a little bit.
Yeah,
But
I think so.
specifically, does it have any transcriptional regulatory role? I think perhaps within the nucleolus, or do you know that if it binds chromatin?
Yeah, we definitely are interested in finding out what genes are affected by stasmin D1. If we have the ideal model and we can knock out stasmin D1 and then see, maybe apply some extracellular stimuli and see what are the changes in the gene expression.
Great. And another question was, would tubulin post-translational modifications affect stasmin D1 binding?
With all the stasmins, they are binding primarily to tubulin dimers. And it is always those tubulin dimers align head to tail in that sequence. So only stasmin 1 has been reported to directly bind to microtubule. And that doesn't seem to be, although like people didn't really investigate a lot about whether PTM changes the binding affinity, but the primary binding partner is not actually microtubule, it's more tubulin. We often don't distinguish the two. We just think the tubulin is the building block, but I think my work has revealed that the tubulin has some other things to do.
Great. Thank you. And then, well, this is relevant to the question you just answered. Do tau proteins displace stathmin D1 from microtubules, assuming stathmin D1 is expressed in neurons?
Stathmin D1 is not a neuronal stathmin. Stathmin 2, 3 are the neuronal stathmin, and stathmin 4 is partially neuronal, partially glia. So most of them, and also stathmin 1, they're I think 1, 2, 3, 4, 1 to 4, they are all like highly expressed in neurons for with unknown function. That's what I'm working on. Stathmin D1 is not. It is mostly in those multiciliated cells, like these in airway tissue, airway, in fallopian tube, in nozzle ciliated epithelial cells.
Great. Thank you very much.
Thank you very much.
I am wondering if there are any additional questions for any of the speakers, you can put them in the Q&A now. If you could, we'll give it a second for that to populate. In the meantime, I just want to thank all the speakers. You guys did a great job, and of course, it's great to see these. Not to take anything away from you, Margaret, but it's great to see these young investigators. The future's bright for molecular cell biology. It was fantastic to see these talks today. I want to emphasize that the breadth of the talks really speaks to the diverse molecular cell biology that is published in MBoC, and it is a real challenge for MBoC to host such breadth. But it's really exciting to see all of this work and the emphasizing, to me personally, it just emphasizes the beauty of cells and cell biology and the molecular underpinnings of, all this complexity that really defines the fundamental unit of life. Really wonderful to see these talks, and very proud to be part of ASCB and MBoC. You can go ahead and put those slides up if there are no other questions. No, there are none. You're wrong, if you wouldn't mind. We could go back to the young investigators, the early career investigators. So I just want to emphasize these early career investigators, and I don't need to say each of the names because I'll probably mess them up. But looking forward to our next series of talks, which will include more young early career awards. And that's going to be, could you remind me of the date of that, please? So I don't mess that up. You're wrong.
October 30. Yeah, I will share later. Yeah, October 30.
Thank you. If you put that link in, that'd be great. Thank you very much. And just a couple of small points. Remind you that what I've learned as being the relatively new editor-in-chief of molecular biology of the cell is it really is a community journal, and we strive as an editorial board to do what we can to support the community. We're here to help you publish your manuscripts and papers in a well-recognized and significant journal. We strive to make sure that we can review papers expeditiously, fairly, and give constructive feedback to the authors so that those papers can be published as quickly as possible. And on the rare occasion when we do delay, it's generally because we have a problem with recruiting reviewers and editors. And so if you have any, if you were asked, we would appreciate your support. Finally, MBoC has a policy of sending all papers that are submitted by ASCB members to send those out for review. And that guaranteed peer review as a member benefit has really, I can tell you that when papers get reviewed, they have a very good chance of getting accepted because of the process that we go through, being trying to be constructive as editors and reviewers. And so encourage you to join ASCB, support our community, come to the ASCB MBO meeting in Philadelphia, and submit your papers to MBoC so that we can help improve the visibility of MBoC and the visibility of young scientists like you've heard today. So thank you very much for attending. And if there are no final comments from you, Wrong, or anything in the Q&A, which I do not see,
Yeah.
I'll bring it to a close. You, Wrong? I'll
Yeah.
let you close it.
Thank you, everyone. Thank you to our speakers. Thank you to our attendees. Thank you.
See you soon. Thank you.
Okay,
Bye-bye.
goodbye.
Thank you, Eric.